Welcome, or wherever your time zone is, a very warm welcome to the History of Science Museum. In the week of our reopening, I am sitting here in front of our festively lit ceremonial entrance. We are so excited to be back, and how better to celebrate that with the event tonight. We haven't been sitting around twiddling thumbs these past few months. We have been very busy developing our very ambitious strategy for our centenary called Vision 2024 with the new vision and mission statement. And as it happens, the mission statement talks about exploring connections between people, science, art and belief, and sharing stories of science in Oxford. Now, what could be more apt than to talk about the huge breakthrough in scientific history with the Oxford AstraZeneca vaccine developed here just a few yards up the road, and to talk equally excitedly about the acquisition of Luke Jerram's glass sculpture of that very Oxford vaccine, which we have just acquired for the museum collection. And why are we doing that? Because we have an initiative here which the History of Science Museum is leading on with the Bodleian just across the road on collecting COVID-19 at Oxford University. And that project, and you hear doubtlessly that I'm virtually jumping up and down in excitement, has just received funding from EPA Cephalosporin. So we will be able to collect all the stories, all the background, the objects, the ephemera, the archives of the work that's been happening here in Oxford. And that is hugely exciting. We will start, we will hear from Sarah and Luke in a moment, but let me do a spot of housekeeping. It's the kind of when I take on my role as a stewardess. Live captions are available. Thank you very much to Samantha from Stage Text for supplying those this evening. If you would like to access the captions, you will find a closed caption icon, CC, at the bottom of your Zoom screen. If you use a computer, it may be slightly different if you use an iPad. We also have a Q&A icon at the bottom of your screen. If you click on that and type in your question, we can group the questions. We will have some time, that's our plan, to have some time at the end of this event for audience questions to come in. We would like to group them so we can make sure that we answer as many questions on a particular topic as possible. My conversation partners tonight, Professor Sarah Gilbert, the Syed Professor of Vaccinology and the Nuttall Department of Medicine here in Oxford, and the lead researcher on the Oxford vaccine team, and Luke Jerram, multidisciplinary artist who is often found working at the intersection of art and science. Very warm welcome to you both. Thank you very much for joining us this evening, or wherever else somebody is in the world, for joining us for this event. It's a great pleasure to have you here, and we are so grateful that you're taking time out of your busy diaries today. So, Sarah, may I start with you? You have become one of the world's most instantly recognizable scientists. Your name is known to people around the world, but that, of course, is not how it started. What drew you to the topic, to this area of research, and what do you hope for students who are studying in this field now?
Thank you. Thanks for the introduction, the opportunity to speak to you all this evening. So it was obviously never my intention to become an internationally known scientist, but what I'm always been interested in doing is understanding how biological systems work, and how we can modify them, and how we can use biology to improve human health, and different aspects of research into medicine, biology, biochemistry, biotechnology I actually came to Oxford to start working on a genetics project, having spent 4 years working in a small biotechnology company, but that very quickly led into vaccine development. So when I came to Oxford, it was to work on the genetics of host parasite interactions in malaria. And that is looking at how the genetics of the human host and the genetics of the parasite work together to shape the course of the disease. But that actually led to the idea of developing a vaccine against malaria, which would in particular work through strong T-cell responses. So most people are familiar with antibodies, which are one of the ways our body has of fighting disease, and they're part of the adaptive immune system. That is, we make antibodies that are specific for different viruses and bacteria and parasites that can cause disease. And we have to generate new ones against all of the different viruses. And we can use vaccines to generate the antibodies, which means that we don't have to have the disease in order to generate immunity. So it's a safe way to protect us against the disease. But there's another part of the adaptive immune system, which is the T-cell response. And what T-cells can do, they're also specific, but they can recognize cells that have been infected with a particular pathogen, a virus, for example, and they can destroy that cell and stop the virus from spreading any further through the body. And they're important in protection against malaria, because what the malaria parasite does when it first gets inside the body after somebody's been bitten by an infected mosquito is it very quickly goes and hides away in a liver cell. And antibodies can't reach it there. And there aren't very many parasites at that point, but they start multiplying in the liver cells. And if we have T cells that can recognize that they're infected, that liver cells are infected with malaria, they can destroy those very small number of liver cells before the infection really gets going and stop the infection from moving on and before anybody becomes ill. So that was how I got into vaccine development. And over the years, working out ways to bring about immune responses in people. And we started working on what we call platform technologies, which are ways of making vaccines that we can apply in lots of different circumstances. And the adenovirus that we use for the COVID vaccine is one of these platform technologies. So I'd worked on that for some time. We started on it in about 2012, I think, first developing the vector and testing it in the lab. And I'd used it to make a vaccine against influenza. And I got interested in vaccines that cause outbreaks. So there are other viruses around the world that we know about that we don't have vaccines for yet. So Ebola is one, and there's a big outbreak of Ebola in West Africa in 2014. And that really got people thinking about the fact that there are these pathogens that sometimes we know about them from causing very small outbreaks, but they can actually cause much larger outbreaks, and we don't have vaccines, and it would be a good thing to develop them. Another one is Nipah virus that causes a hemorrhagic fever also. That's found mainly in Asia. Lassa fever virus, which is found in West Africa. And MERS coronavirus, Middle East respiratory syndrome. So that's found mainly in Saudi Arabia, but there was a big outbreak in South Korea as well. So I've been using the adenovirus to develop vaccines against these different diseases, moving fairly slowly because in research we have to do a little bit of work, generate some data, and then apply for some more funding and move to the next stage. So all of that was going on. And then the WHO encouraged us to start thinking about what they call disease X. And that's the new pathogen that we haven't met yet, that's going to arise, move out of animal species most likely, and start causing infections in humans, and we have to be ready to respond to it. So I've been thinking with my team about how we could use the technology that we had to very quickly develop a vaccine against a new pathogen. And we hadn't really done a very large amount of work in that area, but we've been thinking about how we could move quickly if we ever needed to. So at the beginning of 2020, on the very first day, actually, when I first started to read about a cluster of unusual pneumonia cases of unknown cause, there was the possibility that this is disease X and that we should start making a vaccine. So I started following the story over the next few days. It became clear it was a viral infection, but it wasn't one that we already knew about. It wasn't influenza, for example. It wasn't an adenoviral infection. We then heard that it was a coronavirus, but a new coronavirus, one that hadn't been identified previously. So this then was the opportunity to use the technology that we had and the planning that we've been doing to quickly make a vaccine against this new virus. And I have to say that at the time we started, we were thinking that we don't know if we need this vaccine. We don't know if, at that point, we didn't even know if the disease was spreading from one person to another, although it soon became clear that it was. If it wasn't, then we wouldn't need to develop a vaccine against it. We didn't know how quickly the outbreak would spread, how far it would spread. Would it be controlled within weeks simply by quarantining infected people, in which case we could have started to make a vaccine that nobody wanted. But on the other hand, if we didn't start as soon as possible, we were wasting time. So we decided to start and move as quickly as possible. And as we got more news about the spread of the outbreak and then moving to different countries, we were putting together all the plans for doing the early testing of the vaccine, but then planning to manufacture it ready for clinical trials, because that's a very specialized activity. You can't just make something in the lab and then start vaccinating people. And planning those clinical trials, and it really took off from there.
Well, the response was, of course, breathtaking. I remember watching Andrew Marr show one Sunday morning, and people still talking early last year, people talking about what was happening over there in China, which seemed a long way away. And shortly thereafter, you were on the show talking about the vaccine development. So it was utterly breathtaking for all of us. Do you think that was because you were so prepared as a team for that pathogen X, or was there any particular work you had done that well aligned with this particular virus?
Well, it's absolutely necessary to be prepared in advance for this. We couldn't have just decided that it was interesting in January 2020 and then started on the work. And we also are very lucky that on this research campus in Headington in Oxford, we have the facilities to do all of the early parts of the work. So I could get the first batch of vaccine for the early testing made in my research lab. I have staff who have the capabilities to do that. But we also have a manufacturing facility on site where we can manufacture vaccines for clinical trials under all the highly regulated conditions that we need. And that is very, very unusual in a university. But it meant that we'd already worked out the process to get from the lab into the manufacturing facility in the shortest space of time. We'd worked out how to get the vaccine manufactured as quickly as we possibly could. And we have our own clinical trial center as well. And we've done many early phase clinical trials. You know, the first time a vaccine's ever used in humans, we've done that a lot of times with different vaccines. So we also had very good relationship with the MHRA, the regulators who have to give the approval for the trial to start, and the ethical committees, and the staff here knew exactly what the process had to be, what documents had to be prepared. So this comes after, well, for me, 25 years of research in vaccines, and for many other people here, you know, more than 10 years in many, many cases of doing all of these different aspects of vaccine research, because there's a lot of separate individual things that we need to do and move from one to the other as quickly as possible. And so we had the skills as well as the facilities to get this moving really quickly. And that's how we were able to start the clinical trials on the 23rd of April.
And there was clearly a lot of collaboration going on to develop the vaccine so quickly. Do you think there will be lasting changes to the way in which the global scientific community work together after the pandemic?
I hope that there will. We've certainly learned a lot about how to move more quickly. We've also learned about the things that weren't there when we needed them. So one of the problems that has made life difficult for us is the assays to measure immune responses after vaccination. So there are lots of vaccines, COVID vaccines, going into development around the world, and we need to take blood samples from the people who've been vaccinated and test them to see, have they made an immune response to the coronavirus? And everybody had a slightly different way of doing that. So it meant that we couldn't compare results and know if the immune responses to vaccine A were stronger than the responses to vaccine B, for example, which would have been useful. and how do they compare then to people who've had the disease and recovered from it. And that's still something actually that isn't really put into place now. Another thing that became apparent is the lack of vaccine manufacturing facilities that were ready to start manufacturing in large amounts. So we were very lucky to partner with AstraZeneca quite early in this process, and they decided that they would do all they could to facilitate manufacturing of the vaccine at multiple sites around the world. And they've actually set up supply chains in over 20 different countries, so that the vaccine can be manufactured in different places for supply around the world. But there aren't enough facilities that are able to manufacture to the right standards fast enough. We need more. There's a complete lack of these very highly specialized facilities in Africa, for example, and we would like to see more of those facilities being built and brought into use. So we've learned a lot about what went well. We've also learned about where the gaps are, and we've learned that in a pandemic, if you actually need to vaccinate the world, you're going to need an awful lot of vaccine, and everybody needs to plan for that.
Yeah, I can imagine that. It can't be often that your work is so public. You were on the news all the time. Can you tell us a little bit about the public support you got from very early on, people being so interested and crucially volunteering for trials so quickly?
Yes, we had an excellent response for people volunteering for trials at every stage when we asked for younger people, older people. We never had any difficulty in recruiting the volunteers we needed, and for them coming back and having more blood samples and agreeing, yes, they would have a second vaccination and so on. It's been absolutely wonderful, the support that we've had in all of that. Obviously, you can't test a vaccine unless people will volunteer to take part in those clinical trials. And the support from the public when the vaccine rollout started is excellent as well. And we're really pleased to see that the vaccine's been welcomed so widely. We just wish there was more of it to get to more countries in the world more quickly.
I mean, it may be too early to say, but I'm sure in your group will have discussions. What comes next? How do you look at new variants? How might vaccine technology develop in the future? Or if research showed that the virus affects different sexes or different ethnicities differently, might vaccines become more personalized?
So although there may be minor differences between sexes or between ethnicities, they're really not very large, and there's probably not a need to try to personalize them. And it's actually, in most cases, best to keep things simple and have one vaccine and one way of using the vaccine that works for everybody. Sometimes you might have a different dose for children, but in general, if we can keep it simple, it's more likely to be easy to use the vaccine. On the question of variants, yes, we started from December working out our pipeline to develop vaccines against the new variants. But again, it was work at risk. We started early without knowing if we really need it. And we're still assessing whether we're actually going to need to introduce a new version of the vaccine, and if so, what it should be. But there's a lot of work going on in that field. We're not feeling that we must respond to every new variant that's out there with a new vaccine. We're seeing, as all the other developers are, that actually the immune responses from the first round of vaccines are cross-reacting well with the different variants. So we can probably continue with the existing vaccine with confidence for some time yet.
Thank you. I would like to bring Luke into the discussion, if I may. Luke, if you would kindly come on screen and tell us a bit about your artistic practice and making the COVID-19 vaccine sculpture.
Yeah, well, I'm based in Bristol. I've been working as an artist for maybe 24 years now, something like that. My practice is quite diverse. I'm going to share my screen. I'm going to show you this little presentation that I put together. Hopefully it will work. Yeah, that seems to be good so far. So far, so good. And then we're going to go to slideshow from the beginning. There we go. Does that work? Can everyone see that?
It does.
Good. So yeah, my practice is quite diverse. I do very small things that are quite fun. This is an engagement ring I made for my wife and it's got my voice etched onto the outside of it like a phonograph. So you can play it back on a miniature record player, this one I've made together, and it plays my proposal. It's like a 23-second long message, and that's how I proposed to my wife, with this talking ring. That's kind of crazy, isn't it? A lot of the artworks I made are created in collaboration with people. Here, I'm sort of responsible for putting lots of pianos. If you've ever traveled around or been through train stations or airports, I'd probably put a piano there, a street piano, and they're there for anyone to play. I've installed about 2,000 of them around the city, around the world, about 67 cities, something like that. But now you see pianos in train stations and airports all over the globe. So it's sort of become part of culture. They often get decorated by local artists, and they're there as a blank canvas for everyone else's creativity. Yeah, fun things like this will just transform the high street in Bristol into a giant water slide for the day. I like the idea that a city is a kind of a blank canvas for an artist to think about how we can reimagine our cities. And yeah, there's an element of fun, a sense of play, participation. That's quite important for some of my practice. But then I'm also interested in science and engineering as well. So this artwork is called the Palm Temple. And inside, well, it was created really for Sky Arts. It's at the University of Bristol in their chemistry department. And inside of it, it's sort of mirrored floor, but it's got a bell at the top that rings every time there's an extinction of a species. So there's something like 150 species around the world lost each day. And this bell, it tolls every time there's a loss of a species. And that's a sort of statistic that's come out of the United Nations report. So I'm often, a lot of the artwork I do has a sort of an environmental message, but I'm also thinking about the visualization of science as well. And I'm also touring this giant replica of the moon as well. So this is created from high resolution NASA imagery. And it's 7 meters in diameter. So it's sort of 500,000 times smaller than the real moon. And I'm touring it about. It's going to science museums. It's going to universities, even in swimming pools where you can swim out underneath the moon. It's very beautiful and a lot of fun. And yeah, and we're taking it outdoors to light festivals and museums all over the place. So yeah, if you need a moon, I'm your man. And then we've also got an Earth as well, and this Earth is beautiful. It turns slowly. This is a photograph of it in Greenwich at the Naval College in the Painted Hall. It's very beautiful. That exhibition will be opening again about at the end of this month. So again, this artwork comes with a surround sound composition, but it also acts as a venue for events to take place. So we've had astronomy lectures underneath the moon, and we've had choirs performing and yoga lessons and all sorts of crazy stuff. So yeah, a lot of the artwork, I'm interested in science. I'm interested in, because I mean, it's all the breakthroughs in society that they seem to be happening in science, you know, and so it makes sense for an artist to respond to that. But I'm also interested in how things work, and I was sort of trained in engineering and science, a little bit in physics. So yeah, a lot of the work involves science. Now, these are all images of and diagrams of HIV. And this leads us into this body of work that I've been making over sort of 14 years, these glass virus sculptures. Now you can see they're all rather different, and yet they're all of HIV. And I'm colorblind, so I've got a real interest in perception and in the process of seeing, but also in the visualization of science. And what I noticed is that a lot of these colors, some of them are artificially added. So the idea for this project came from looking through a newspaper and noticing an image of, say, HIV used to talk about the pandemic in, or AIDS in Africa, whatever. But they'd be using an electron microscope image, and then the color had been added. And sometimes the color is added for scientific reasons, to highlight a particular protein, but other times it's added for just to add some sort of emotional impact. And I had a problem with that. So I decided to create my own version. This was back in 2004. And I created this glass sculpture about the size of a cricket ball. And as a sort of object to contemplate really what HIV was. And it led to this body of work that has been made over a period of since 2004, maybe 15 years now, with my team of glassblowers, and we work with virologists and scientists as well to create these glass sculptures, and they're now in museums all over the world. The photographs even get used in medical textbooks as well, so you see them in journals and textbooks. And when you make an artwork, you're never entirely sure what the outcome will be and where it will go, and that's quite exciting. So yeah, it was just an unexpected outcome. So that the photographs of these sculptures have become part of the language of virology. And you're obviously attracted to them. The sculpture themselves are about sort of this big, but you're also slightly repelled by them as well, by what they represent. So the sculpture you've got in front of you is one of smallpox. So this virus has killed more people than any other virus in human history. But at the same time, it's something to be. This is something that humanity has dealt with through vaccination. We've managed to globally eradicate smallpox. And it was at the beginning of 2020 that Duke University over in America invited me to make something, a coronavirus, because at that point the pandemic was, well, for them it was right at the very, very beginning of their and they were already sort of researching coronaviruses. And they asked me to make a sculpture. And then it was only a few months later, when the artwork was completed, that I'd realized the impact of what this thing was that I'd suddenly made. Because at that point, the whole world seemed to be turned upside down. And all the press came to my studio to take photographs of this sculpture. And suddenly I had lots of inquiries, people wanting to buy them. So we've sold quite a few now, and we've raised, I don't know, maybe about £50,000 for Médecins Sans Frontières. And that's been really useful for them to buy inflatable hospitals to help communities impacted by the pandemic. We've made a larger version now as well. And then, yeah, I went down with coronavirus myself in November. In fact, I've still got tinnitus now, and yeah, it's not good. And it was at that point I thought I should probably make an Oxford AstraZeneca vaccine to help kind of communicate perhaps what the vaccine looks like, and perhaps it might be useful to help get people on board to get vaccinated. And there's a little film of it here turning. And I think maybe I think you're going to maybe present it turning, perhaps, hopefully not as fast as this. I'm slightly scared to see this on display turning around, but we'll see what happens. And then finally, I was going to also show you how these artworks are made. They're made with borosilicate glass. So this is the same type of glass that you use to make distilleries and test tubes. Now, how do we get out of this? Oh, I've done it now. I'm going to stop that. I'm going to start again. I'm going to, I don't know, that one there. Yeah, I was going to show you this little film. It's worth looking at. Hopefully that's going to work. So I've been working with this team of glassblowers for about 15 years. And yes, the same skills used to make distilleries and test tubes. You can see here we've got the genetic material, the RNA or the DNA, and that's put into the bottom of a glass flask. The whole thing has to be kept hot for quite a long period of time. It's on a lathe, and then it has to be sealed in, basically, to turn what you see there into a sphere. So there's not many people in the UK that can do this. There used to be a glass department in more or less every university, chemistry department, sort of knocking out glassware. But now a lot of the glass is manufactured in China, and a lot of those skills are being sort of lost. So once it's all sealed in, then all the external proteins are then all handmade. You can see the diagrams that I sent to my glass team over there. Diagrams often signed off by scientists, you know, so they're as accurate as they can be within the limitations of glass blowing. So all these are handmade, and then finally they're fixed onto the outside of the sculpture. You get a sense of it anyway. There we go. Just finishing off a little tree. So it does take a while, and then you've got to fit them all on. Okay. I think that'll do. Stop share. There we go. How was that?
Thank you very much, Luke. That was utterly stunning, and I think we might well need a moon. We've got the Russell pastel of the moon, of course, would look really lovely juxtaposed with one of your artworks. But let's concentrate on before I put in my order. This is clearly a very personal response to the pandemic. What was important to you as an artist to respond so fast to a global event? Was it your personal story of having contracted the virus? Or do you feel in other contexts as well that you really need to respond to what's happening out there?
Yeah, I suppose there is I mean, it's taken sort of 12 to 14 years to build this body of glass artwork. Mm-hmm. So that we got a sudden momentum. It's similar to the development of all the science. All that research is there. So as soon as it happened, in fact, the artwork was already almost in production before we realized it was a pandemic, you know. So it kind of made sense to create both of those works. And also, I think if everyone can do what they can during a pandemic, whether that's helping a neighbor or dealing with the science or helping with the vaccinations, then that's brilliant. And the only thing I could do was carry on working as an artist, really, and helping to create artworks that would perhaps help visualize what the problem was.
Mm. And we're delighted, of course, that as far as we know, the sculpture, the History of Science Museum, is the only one in the public institutions. The others are in private hands. So if you want to see the sculpture, come and look at it in Oxford. Can you say a little bit more about your broader glass microbiology, the body of work you're doing? Why did you choose glass sculpture to explore the microscopic world of viruses?
Yeah, I suppose it's about the fact that these glass objects, they're colorless. So it's that mm-hmm. response to looking at colored electron microscope images of viruses and realizing that, you know, viruses don't really have color because they're sort of smaller than the wavelength of light itself. So I wanted to create an alternative visualization to help communicate what viruses were.
You've shown us some beautiful examples of your work, and it's obviously incredibly varied, but there appears to be a common thread that intersects with science. What draws you to explore the ideas of science?
I suppose, yeah, I'm just interested in how the world works. I was one of those kids that would take my parents' radio apart and then to find out what was going on inside. I think a lot of scientists are a bit like that as well, that they're the people who are interested in asking questions about the world. And like scientists, but, you know, I think artists just have a different set of answers perhaps to the same questions. Yeah, the radio would never seem to work properly after I'd put it back together.
That reminds me of an alarm clock I tried about as a child. Let's take it as an offline discussion what happened to that. As I said at the beginning, our new mission at the museum is to explore the connections between people, art, and science. In your response, or in the way people respond to your work, have you learned anything that connects your work to science or the response to science?
Yeah, I suppose. I mean, these days I often get approached by scientists to help communicate their research. So sort of 30 years ago, it felt like sort of scientists could hide in their ivory towers a little bit and get on with the work. Whereas now it's almost part of their funding that they have to be able to communicate what they're doing to the general public, both to sort of help communicate that research, but also even to bring in students as well. So I'm often working with universities to help them with that. But it was interesting when I presented the moon at the Natural History Museum, we had massive audience numbers, right? 2.1 million visitors came to the museum. And some of those kids seeing the moon will be really switched on by it and will want to go and study space science as a consequence. There was a little girl who came up to me and she said, "Will you put the moon back afterwards?" And I thought that was really cute. But at the same time, I met a scientist from the European Space Agency, and he'd studied the moon his entire life as a scientist, his entire career at least, and he was in tears. And he'd seen the moon in front of him for the first time as a three-dimensional object, and he became incredibly emotional, and he needed a hug. So I had to give him a hug, and that seemed to sort him out.
Thank you. I would like to bring Sarah back into the discussion at this point. The point you just made, Luke, about scientists reaching out, there's a lot of engagement with the public, of course. And of course, Sarah, there are colleagues in your department who focus on that. Do you both see opportunities for scientists and artists to work together going forward, more than what was common in the past, having a kind of artist in residence concept, which is very common, but sometimes strikes me as somebody there observing? That's not really collaboration. How do you feel about it? Sarah, may I start with you?
Yeah, I think it's really interesting for the public to see these representations of the viruses. And it's interesting for scientists as well, because although we kind of know what they look like, we've seen all the electron micrographs and so on, we don't actually get to see them in real life. We're kind of working blind most of the time because they're so tiny. We end up working with very, very small amounts of colorless liquid in a tube, and then adding it to another amount of colorless liquid in another tube. And what we're actually doing in some cases is creating life, because we're taking something inert, a piece of DNA, and then the DNA has the whole genome of the virus, and we can manipulate that and turn it into a vaccine. But it's just a piece of DNA, and then we'll put it into cells which are big enough to see down an ordinary microscope, and then the DNA starts to turn itself into a virus, and we've created a new life form by doing that. But then when we purify it, it's again too small to see. So we're kind of relying on various techniques to detect its presence and to sequence the DNA, to read the sequence and see if we've made what we intended to make, that there are no mistakes in it, that it's a faithful representation of, for example, the spike protein from the coronavirus, and that it's all present and it doesn't kind of drop out halfway, something like that. But we're doing all of that without really, well, we can't ever see the virus. And even if we could see it, it wouldn't answer the questions that we have about it. So we're working in a different way with the knowledge of what's going on mainly in our heads, not being able to look at it. So it's a very different thing to be able to see the whole virus and then to be able to talk about the different parts of it and what they do, where the genetic information is carried and how it interacts with the cell and so on. And I think that helps people understand what it is that we're doing. I think one good thing that's come out of this year is that maybe people will have a much better understanding of the fact that there are lots of different ways of making vaccines. There are different types of vaccines. They're not all the same. So some people haven't taken much interest in vaccines before and like to dismiss vaccines. They contain mercury, they contain aluminium. Well, actually, let's talk about which vaccine do you mean? And no, they don't contain mercury. And different viruses, different vaccines may have had different components in the past for various reasons, but the ones we're making are actually live, but they can't spread in the body. There's lots of different ways of doing this, and I think maybe people have got a better understanding now of the different ways. I mean, the flu vaccines are made by producing lots of flu virus and then breaking it up with detergent and using part of the virus itself, but it's not the whole virus. We're using a whole virus, but it's a virus that's been modified so it can't spread. So it's quite complicated, but maybe by looking at these visual representations, we can help explain that more fully to people.
Mm-hmm. Thank you. Luke, what's your hope for the future of collaborating with scientists? What do you think are what can still be out there, what we haven't even tapped into yet?
Yeah, it is amazing, isn't it? I mean, the pandemic has created sort of amateur virologists out of 60 million people, and the entire world now is fascinated in virology in a way that they never were before. It is an extraordinary thing, and everyone seems to have opinions and ideas and really interested in it, not just in terms of health, but they're passionate, and it's an extraordinary thing. And so in terms of the education of our country, I mean, it's amazing, isn't it? I mean, I'm very lucky, I suppose, as an artist. I get to hop around, so I can go and collaborate with a virologist one week, and then I'm able to go and speak with astronomers the next. So there's a certain pleasure in that. The rules of engagement of art and science are very different. There's a set of rules that you have to follow in order to do science properly. And the rules of art, there are some rules, but they're just quite different. So collaborating with artists and science collaborating is quite tricky, because you often end up with outcomes that either fulfill the scientific brief or they fulfill the artistic brief. But something that does both is quite tricky. And I've learned that through sort of 20 years of sort of making art. But I think there's still a role for artists. And I think just as another method of helping to communicate the research that is being done and helping to perhaps break some very complicated ideas down into something that can be visualized, because the public, I think, can understand science and they can understand phenomena in different ways. Some people are very visual, and some people need a model, so other people need a diagram, some people want a radio program about it, some people want to read an academic journal. So I think if we can provide alternative methods of communication, then we can reach more people and have a larger impact.
And thank you. Most definitely also show the beauty of science, of course, which is often not thought about. Science is something which exists in the lab, but that science can be so utterly stunning is something which many people have really responded to over the past year. And of course, also the visualization of aspects of our life, which may in the past have been seen as something horrible or ugly, even like.
Yeah. Well, I always like to think that actually, viruses aren't science. Viruses are naturally occurring phenomena in the same way that space and planets and stars, that they're not science or astronomy either. They just exist as things. Science is a way to study something, and it doesn't, the stars don't belong to scientists. They belong to everyone.
Well, that is, of course, can I quote that for our next what the museum is about, please? Because we are talking about science and belief systems are different ways of making sense of the world around us, trying to understand what the world is about. But Sarah, may I get back to you and just ask, did you ever imagine that artists would be responding to your vaccine research like this, making sculptures of it?
No, this was something very new for me. We have had people working in the media team creating cartoons, comics to help communicate with school children, but you know, glass sculptures, that's something that I wasn't expecting, but they're very beautiful. But you know, we do have kind of slightly mixed feelings about them, because some of these viruses can cause us harm. But then other ones can help us. So I think it helps just to have an understanding of it. It's a piece of nature. It's a type of life form, and we should maybe just try to understand how it works instead of worrying about whether it's a friend or foe.
Yes. Am I right in thinking that viruses are necessary in order to help our immune system? Is there a sort of, they're required to help boost our immune system as a survival mechanism, or am I wrong about that?
Well, our immune system responds to viruses, but if the viruses weren't there, we wouldn't have to fight them off. So
fair enough.
Yeah.
And Luke, just to look at your work again, are there any areas of science that that haven't been explored yet, that you haven't explored yet, rather, that you would like to get into?
Yeah, well, I mean, we're facing a sort of environmental crisis, aren't we, really? And I'd be keen to kind of collaborate with scientists to help in any way I can to help with that environmental and crisis, whether it's about extracting carbon dioxide from the atmosphere or anything else. Yeah, I like to think I'm kind of creative problem solver, and maybe, you know, if I might just quit the art and go into science and engineering and see if I can actually have some real positive impact on the world. It might be more useful for society. But yeah, I mean, I'm interested in all sorts of things, and I'm happy to apply my mad way of thinking to help in all sorts of different ways.
Well, I'm sure we will all benefit from whatever you choose to do next. In terms of your artistic work, do you come up with the idea first and then work with the researcher, or do you find a researcher, do you find a scientist and collaborate with them, and then the outcome is the artwork, or does it entirely depend?
It's a whole variety of different things. Sometimes I'll come up with ideas and they'll just sit in a pile. I've got this big pile of ideas, and then 2 years later someone will come along and say, I'm looking for an artwork to go on top of a building or something, and there'll be something there ready to go. Other times, institutions or organizations or festivals or museums, they contact me and say, look, we're looking for something that relates to climate change. We've got a festival in July and we've got a budget of X. What can we do? And I'll generate some ideas to sort of fit the brief. And so, yeah, I'm working with all sorts of different organizations around the world to help generate ideas. And sometimes they thought that they just, they never get taken up. Sometimes, so for like English Heritage, they contacted me. They saw the giant water slide down the street and said, look, we've got all these castles and we want you to animate them in interesting, fun, exciting ways. So I come up with some fun and exciting ways. And they said, this is exactly what we need. These artistic ideas are absolutely perfect. But we're just a bit nervous as an institution. You know, this is English Heritage. I think we'll just carry on doing our historical reenactments after all. You know, so I end up with this big stack of ideas that I've never taken up, but hopefully I'll get through them one day.
Well, we all look forward to that, I'm sure. I would like to bring in some questions from the audience that have been pouring in. Sarah, people would like to know what the biggest unknown in your field of research is, and that you would be really excited to find out more about. Big question, that one.
Okay, the biggest unknown. So the biggest unknown, when it's a virus that's new and it's not very similar to a virus that we've already got a vaccine for, is understanding what the immune response has to achieve to protect ourselves against it. So we know that we can give somebody a vaccine and they make some antibodies and their T cells get a bit stimulated. What we need to find out is how big does that immune response need to be? What level of antibodies do they need to protect them? And that's something that we haven't known about coronaviruses, because although there are 4 coronaviruses that circulate around the world every year, and they give us a cold, and we don't know that it's a coronavirus. It could be flu, it could be an adenovirus infection, but we have a cold and feel a bit poorly for a few days. Some of those are coronaviruses. We don't have vaccines against any of them. And if we did have, it would have been very helpful this year in understanding that we need actually quite a high level of antibodies to protect us against this novel coronavirus of SARS-CoV-2, as it came to be known. So that's one aspect of that. That's a big unknown for each pathogen that we need to protect ourselves against. What type of immune response directed against which part of the virus? How strong does it need to be? It's not enough just to say that we've developed some immunity against a virus. Is it really strong enough to protect us, and how long will it last, and all of those kinds of questions. So there's still a lot of work to be done in that area, because we don't have a full understanding of that.
Thank you. Luke, there is a lot of fascination amongst our audience members about the production, the way the glass sculptures are being made, the video you showed us. Can you say a bit more about the collaborative practice with people who have different skills in creating your artwork?
Yeah, so I often come up with an idea, and then I've got to work out how it's going to be built. And I'll find specialists, whether it's composers or architects or engineers, even hot air balloon manufacturers. And then my job is to then learn the language of that new discipline in order to have the conversations with that fabricator, to learn the language and work out what's possible and what isn't. And so it's about learning those it's a new method of communicating, really. And then I'll do diagrams and work carefully, and we'll do prototyping and developing different ideas before production begins. And it keeps my life really interesting. By collaborating with all these different specialists, I can make all sorts of different artwork that physically I couldn't personally make myself. And it also means I'm not sure what sort of artwork I'll be making in 10 years' time as well. So it's through collaboration. It's the same with science, that anything's kind of possible. And
to follow up on that straight away, you, Sarah, our audience members have expanded that question. I would like to hear about your collaboration, not just within your team and researchers in the field around the world, but also with researchers from very different fields. Can you elaborate on that a bit?
Well, we've been talking to people this year who work on following the virus, looking at how it changes, sequencing the virus. That gives us information that we need to understand the new variants that are arising. Structural biologists who work on how proteins are shaped and how viruses are assembled from different proteins. We've worked with those as well. And then there's a lot of regulatory science. So there's a lot of stuff that rarely gets talked about, actually. And there's a huge amount of very careful, detailed work that goes into not only making a vaccine in a very controlled way, but testing it and understanding exactly what we have in that vaccine vial before we attempt to vaccinate anybody with it. And that we've got a glass vial with a colorless solution in it, and we need to know an awful lot about that before we're confident to give it to people. And developing the ways of testing it and understanding exactly what's there is something that's quite complex, and then also has to be presented in a very detailed way to the regulators who will assess it and decide if we've got something that's suitable to use.
Thank you. Another follow-up question here from the audience. You talked about, Sarah, large-scale lessons that were learned during your work on COVID-19. But people wonder whether there were also small-scale lessons, perhaps in the process of creating the vaccine, that really surprised you.
Well, not so much in the process of creating the vaccine. We kind of, we have our plans and we put them into place. And we have to know what we're doing. We're not just it's not just kind of suck it and see. It's follow an approved process to generate something that so that we know what we have. I think a lot of the lessons that we learned were really just to do with organisation and logistics in getting everything in the right place at the right time. We had to put up 2 mobile vaccine clinics next to our vaccine centre because we had to respond with clinical trials against this pandemic virus during a pandemic. So we had to implement social distancing in our vaccine clinics, which was something we never expected to have to do. We had to work out how to make it safe for volunteers to come into the clinic and move around the site, and we needed a lot more space to do that. So we learned about how to put up mobile buildings very quickly. We learned about how to get vaccines from Italy when there are no flights because there's a pandemic, which the answer is charter a plane. And there were a lot of it was just organisation and making sure that things got to the right place at the right time and that all the boxes were ticked and that nothing had been missed out. There's a very complex system of designing the label for the vaccine and getting it onto the vial, and we spent an awful lot of time just talking about labels.
That is, of course, a discussion which is very, very close to our heart here. Talking about labels and what do we put on them. Thank you. Questions keep pouring in. We are beginning to run out of time, so I would just like to finish off with almost the same question for both of you, which the audience have been asking. Luke, what is next for you?
Next, I'm doing something completely different, which is very nice. I've got in fact, I've got a diagram here. I don't know if you can see that. I've got an entire bridge in North Wales that I'm wrapping in giant patchwork, which is quite fun. It'll be very beautiful. So it's quite nice to have that. I'm also touring a giant sculpture that consists of 120 flags made of NHS bed sheets. And so it's a sort of temporary memorial to victims of COVID and a tribute to all the NHS workers. So that is touring the UK this summer. It's going to Harrogate next, and then Edinburgh Science Festival, and then it's going to Weston-super-Mare, I think. But yeah, so it's always quite a large artwork. You can find the information on my website, lukejerram.com.
Which of course we will follow up straight away, and fascinated by the wrapping of the bridge. Of course, I vividly remember the wrapping of the Reichstag. Wow, let me know when that is out. Sarah, same question for you from the audience. What are you going to work on next? Is this mostly focusing on COVID or coronavirus, or is there something else you're planning to do as well, also?
Well, really, I'm planning to go back to what I was working on before the pandemic, which was the development of a number of vaccines against viruses that we already know and that have caused outbreaks and have the potential to cause more outbreaks, and hoping now to apply some of the learnings from the COVID vaccine development, in particular about how to get it all speeded up, because those programs could do with being brought to a conclusion more rapidly now. They've been on the back burner for a while, and I'm hoping that now we can go back and really get those moving and make some more vaccines against diseases which we don't expect to vaccinate everybody with. But if we had the knowledge that this vaccine works against Nipah virus, for example, and we have a stockpile of it, if that starts to cause disease in Bangladesh or India, which is where you often see the cases, you would have a stockpile of vaccine ready to go. And you might, in the countries that have the most cases, vaccinate some healthcare workers so that if they have to go and respond to a small outbreak, they're already protected. So that would be a vaccine to be used on a very different scale, but still very important to have, because it could actually be very useful in responding to outbreaks.
Well, that would be fantastic, of course, to be able to do that, and hopefully the experience with the pandemic now and how matters can be accelerated will help with that. That would be a fantastic outcome amongst the many others. We now need to bring matters to a close. So thank you both very, very much for your time this evening and for the fascinating discussion. I've really enjoyed what you've shared with us, and I hope members of the audience have done so as well. We have a very quick screen poll which we would like to share with members of the audience. I'm sure there are lots of thoughts there, which is coming up for you now. Please fill that in. We'd also like to thank our funders and individual donors, of course, who supported not just the program, not just the program of the museum generally, but the first display of Luke's sculpture and other experimental program we are doing in the context of our centenary preparations, where we want to completely transform the museum. And also, thank you all for joining us for this event. Thank you for your support and interest in events like that. Thank you for coming. Goodbye.